genitourinary system, GIT, and in all those systems there are some cells, there are some specific organs. Likewise, the immune system also has some cells, some organs, but the cells of the immune system, they are not only confined to one particular organ or one particular system. These cells, they are found almost everywhere in all the systems. In GIT also you can find the systems of immune cells of immune system. In a nervous system also you can find the immune cells. So these cells are present almost everywhere. Few areas are there where they are not present, but otherwise they are present almost everywhere. Despite that, there are certain organs, specific organs, dedicated organs for the immune system. If we talk about the structural anatomy of the immune system, there are two types of cell in immune system. One, lymphoid cells, lymphocytes and plasma cells, and others are phagocytic cells. Lymphocytes are responsible for specific immune response, both humoral and cytotoxic immune response. The T lymphocytes are mainly responsible for cytotoxic immunity, and the B lymphocytes and plasma cells, they are responsible for humoral immunity. And the other cells of the immune systems are phagocytic cells, and they are polymorphonuclear leukocytes and macrophages and monocytes. They have a scavenger function. They eliminate old, effete cells and foreign particles from the body. And together they are called lympho-reticular cells, or the immune cells, in simple words. They all are immune cells. So they are of two types, lymphoid cells and phagocytic cells. If we talk about the functional anatomy of the immune system, how does the immune system function? In that respect, we can divide it into two parts: humoral or antibody-mediated immunity, or cellular or cell-mediated immunity. Cell-mediated immunity and humoral immunity. Humoral immunity means that is mainly based on the antibodies produced by B cells, plasma cells, and cellular immunity, that is the cell-mediated immunity, T lymphocytes. So this we're gonna talk about in another lecture, that is the immune response. Today we are going to talk about the structure of the immune system. So the organs and tissues of the immune system, there are two types of organs. One is primary organs or tissues, and the second is secondary organ or the tissues. Primary organs are where the immune cells, they mature. The immature lymphocytes will mature in these organs or tissues, like thymus and bone marrow. So thymus and bone marrow, they are primary organs, primary immune organs, and secondary immune organs are lymph nodes, spleen, various mucosa associated lymphoid tissue, like gut associated lymphoid tissue or skin associated lymphoid tissue. So we can divide the all immune organs into primary and secondary organs. Primary or central, they are thymus and bone marrow, and secondary are lymph node, spleen, and various other mucosa associated lymphoid tissue. Thymus is a gland which is found in thorax, just in front of the heart, and it has got a capsule, outer cortex, and inner cortex, and medulla. And this is the gland or immune organ which is responsible mainly for the development of T lymphocytes or T cell development and their maturation. What happens when the precursor cells from the bone marrow, they reach to the thymus, At this time, these cells are not differentiated. You can't say whether this is a B cell or a T cell, B lymphocyte or T lymphocyte. They are same. They have common markers on their surfaces, and when these cells reach in the thymus outer cortex, they proliferate, they mature, and they acquire T cell specific markers uh, and T cell receptors on their surface. This happens in the outer cortex of the thymus. And then these cells move to the inner cortex. Just beneath the outer cortex, there is inner cortex in thymus, and these cells move to inner cortex, and something interesting happens there. 90% of these cells, these mature proliferated T cells, which have acquired all the surface markers, T cell surface markers, they die in the inner cortex. And why does this happens? It is said that these dying cells are those which are stimulated by the self antigen. I mean, body's own antigens. So these cells, these were the cells which were recognizing the body's own antigen, and those cells are killed in the inner cortex of the thymus, and this is responsible for the immune tolerance, because all the cells which were reactive to the self antigen, they have been killed in the thymus. And then 10% remaining T cells, they move into the medulla, and these are the mature T dependent lymphocytes, or we call them T lymphocytes. This happens in the thymus with the help of some hormones, thymopoietin and thymosin. So the thymus has a very crucial importance in immune system, because there is maturation of T lymphocyte happens in thymus. In fact, we call them T lymphocytes because they are thymus dependent lymphocytes. We call them T lymphocytes. If we cut the thymus of a mouse, so there is dramatic decrease in circulating lymphocyte in absence, and there is absence of cell mediated immunity. Or there is a congenital disease in humans, congenital birth defect in humans, that is called D. George syndrome, in which the thymus fails to develop. In this also, in both these cases, there is absence of circulating T cells and cell mediated immunity, and increase in infectious diseases, particularly the viral diseases. So that is the importance of thymus. The second central organ is bone marrow. In bone marrow, the remaining lymphocytes, they mature in bone marrow. They mature in develop B lymphocytes in bone marrow, and this B comes from bone marrow in humans. In birds, this B comes from some bursa, and so here there is a selection process of the same selection process happens as it happened in thymus. Lymphocytes, B lymphocytes, which are self-reactive, they are either killed or their receptors are transformed in bone marrow. Now coming to the peripheral lymphoid organs, the most important and most abundant peripheral lymphoid organ is lymph nodes. Lymph nodes are present in all over the body. They are encapsulated bean-shaped structure. They contain a reticular network, and they are packed with lymphocyte, macrophagic, dendritic cells. And this is the place where the lymphocyte or other cells, they encounter the antigen. Right? In the central organs, the B cells and T cells, they only mature in the central organs. They do not encounter antigen there. So they are non-antigen specific cells. They are mature cells, but they are not specific to any antigen, both T cell and B cells in the central organs. In peripheral organs, they become specific for antigens. So one of the peripheral organ is lymph node, and so here the cells, T lymphocytes and B lymphocytes, both of them, they encounter antigen and they become specific to that antigen and become effector cells. This is a general structure of a lymph node. It is bean shaped. There is a hilus through which blood vessels go inside, and there is a capsule, and there is outermost layer that is called cortex, and beneath that cortex there is paracortex, and inside there is medulla. This pink color or orange color is medulla. This gray color is paracortex. This blue color is cortex, and within the cortex there are follicles, primary follicles and secondary follicles. Secondary follicles have a germinal center, and in these follicles the lymphocytes, after encountering the antigen, they proliferate. They become specific to the antigen and they proliferate in these germinal centers or follicles. And besides this, the lymph node, it receives lymph through incoming lymph vessels, and there are outgoing lymph vessels through which the lymph will go to the lymph channel and it will go into the circulation. So the cortex, it contains lymphocytes, mostly B cells, and they are present in primary follicles. Once they are encountered with antigen, they start multiplying, they start proliferating, and this primary follicle convert into the secondary follicle, which has a germination center. And the paracortex, it mainly contains T cell area. This is, or it is also called thymus dependent area. In this area, the T cells, they proliferate. And the medulla, it consists both T cells and B cells. They are arranged in branching bands. We call them medullary cords. So the lymph node has cortex, paracortex, and medulla. Cortex is primarily the area for B lymphocytes. Paracortex is primarily the area for T lymphocytes, for the proliferation of T lymphocytes, and it is also called thymus dependent area. The lymph node, what is the function of lymph node, or what is the importance of lymph node? First is the filter of lymph. Lymph from the whole body, they go to the lymph nodes and to the lymph channel, and then again it come back to the circulation. From circulation, it goes into the extracellular fluid. It becomes extracellular fluids, and then it goes to the lymph node again, and this keeps on going, the circulation of lymph. And they are also a place for phagocytic activity of the cells, and there is, as I said, proliferation and circulation of T and B cells. It happens here. And whenever there is an infection, the lymph nodes which are receiving the lymph from that area of infection, they become enlarged, painful and enlarged. And by this we can know there is some problem in the area. It can be an infection, it can be a malignancy. So they become enlarged if there is some kind of problem in their receiving area. The second peripheral organ, second important peripheral organ is spleen. Right. Spleen has, it is present in the abdomen, as I think you know, and it is somewhat similar to lymph node, besides the fact that it has got another area called red pulp. You can it is divided into two parts. The compartment of the spleen are divided into two parts: white pulp and red pulp. White pulp is functionally similar to the lymph nodes. It has proliferative areas for the proliferation of lymphocytes. Red pulp, on the other hand, has RBCs and macrophages. So the white pulp, it is surrounding the branch of splenic artery or arteriole, and Just adjacent to the arteriole, there is an area called periarteriolar lymphoid sheath or PALS. This is mainly populated by T lymphocytes, and surrounding that PALS, there is an area for B lymphocytes or follicles. And these follicles, just like in it happen in lymph nodes, there are primary follicles and secondary follicles containing the germinal centers. If I show you the picture, this is arteriole, a branch coming from the splenic artery, and just adjacent to this arteriole there is a T cell zone, or we call it periarteriolar lymphoid sheath or PALS, and just adjacent to that area there are B cell zone or follicles. This is the white pulp. This area is the white pulp of the spleen, right? Like this. This area is the white pulp of the spleen, and this is red pulp of the spleen. So in this white pulp there is a T cell area which is called PALS or periarterial lymphoid, and these are B cell areas. And what is happening in this? That is exactly almost same what is happening in the lymph node. The lymphocytes, they encounter the antigen and they become specific to the antigen, then they proliferate. That's it. And what happens in the red pulp? Red pulp is actually is function as the graveyard of blood cells, all blood cells, old Ephes, RBCs, and platelets. They all are buried here, and their heme and etc. that is extracted from there. And another function of a spleen is it mount immune response by proliferation of the lymphocytes, and it also filters the blood by trapping blood-borne microorganisms and antigens. So these are three main functions of the spleen. One is it is a graveyard of blood cells, and mounting of immune response and filtration of the blood by trapping blood-borne microorganisms. So we talked about lymph node, spleen, and now coming to the mucosa-associated lymphoid tissue or MALT. MALT is again similar to the lymph node and white pulp of the spleen, but they are present associated with the mucosa. They can be loosely organized, like they are present in the intestine, or they can be well organized, like tonsils, appendix, and Peyer's patches. So loosely organized lymphoid cells, they are present in the lamina propria of the intestinal wall, and well organized mucosa-associated lymphoid tissue like tonsil, appendix, Peyer's patches. And the function is same. That is encounter the antigen or pathogen and develop the antigen specific lymphocytes, proliferate them, like that. And they are present in all the areas where there is direct encounter of the pathogen, like gut or respiratory system or skin or mucous membranes, and they have different name, like gut associated lymphoid tissue is called GALT, bronchial associated lymphoid tissue are called BALT. Bronchial associated lymphoid tissue are relatively less well organized, but lymphoid cells are present there. This is the diagram showing the mucosa associated lymphoid tissue in the intestine. There are germinal center and proliferation area, just like that happened in spleen and lymph node. This is the palatine tonsil. In structure, it also looks like a lymph node or a spleen. So these were the peripheral or lymphoid organs. So we talked about the central lymphoid organs and peripheral lymphoid organs. Central lymphoid organs are thymus and bone marrow, and peripheral lymphoid organs are lymph nodes, spleen, and or MALT, mucosa-associated lymphoid tissue. Now talking about the cells of the immune system. Cells of the immune system, or they are called lymphoreticular system. These cells, lymphoid cells, they are also called leukocytes or white blood cells. They originate from pluripotent stem cell in fetal liver. and bone marrow, and then they migrate to the body sites like bone marrow and thymus. Then they undergo development and they perform various functions. So first we'll talk about the lymphocytes. They are 20 to 40% of the body's white blood cells. So in blood, 20 to 40% cells are lymphocytes, and in lymph, 99% cells are lymphocytes, and they continually circulate in blood and lymph, and they are capable of migrating into the tissue space and lymphoid organs. They can be there are three populations: B cells, T cells, and natural killer cells. When they are present in blood, you cannot tell the difference whether this lymphocyte is B cell or T cell. I mean, on the basis of microscopy. For example, if in this picture, this is the lymphocyte in a peripheral smear. So we can just say it is a lymphocyte. We cannot say whether it is B lymphocyte or T lymphocyte because they look exactly same. But there are methods by which we can differentiate between the two, and they have entirely different function, but they appear exactly same, right? So there are three types of lymphocytes: B cells, T cells, and natural killer cells. On the basis of their life, they are divided into short-lived and long-lived lymphocytes. Short-lived are the actual effector cells. They live for 2 weeks. And long-lived lymphocytes, their lifespan is around 3 years or more, sometimes even for life. They are called memory cells. Memory cells. They remain alive for a very long time, and whenever the body faces the similar kind of incident, similar kind of infection again, they become active and proliferate. Now, as I was saying, on morphology, on light microscopy, T and B cells, they look very similar, but they are different. The T cells, they have thymus-specific antigen on their surfaces. For example, thymus TCR receptor, CD4, CD8, and various other cluster of differentiation antigens on their surface. B cells, on the other hand, they have other receptors on their surfaces. T cell receptor is present on the T cells, although it is similar to immunoglobulins, but on B cells we have B cell receptor, which is an immunoglobulin, right? These are the antigenic difference. If we add sheep RBCs to the lymphocytes, the human T lymphocyte will bind to these sheep RBCs and they will form a rosette. The B lymphocyte will not form this rosette because these sheep RBCs, they have a receptor that bind to the CD2 molecule that is present on the T cells and it is not present on the B cells. So these T cells, these are sheep RBCs, they will surround the T cells because they will bind to the CD2 molecule and they will form a rosette. This happens only with T cells, not with B cells. Similar type of incident can also happen with B cells, but provided if we tag the sheep RBCs with antibody and complement, because B cells, they have a complement receptor on their surface, CR2. So if sheep RBCs are tagged with antibody and complement, they can also give this type of picture, and we call that EAC rosette. E for erythrocyte, A for antibody, C for complement, EAC rosette. Here S RBC means sheep RBCs. So B cells shows EAC rosette and T cells show S RBC rosette. So T cells will show a rosette, S RBC rosette, that is without anything, they will bind to the T cells, and B cells will bind only those RBCs which are tagged with antibody and complement because they have complement receptor on their surface. Another difference is electron microscopic difference, because B cells on their surface have the microvilli. These microvilli structure on their surfaces, they are present only with B cells, not on the T cells. They are present on B cell extensively, and T cells are free from such structures. But this can be seen only under electron microscope, scanning electron microscope, not under the light microscope. Right. Another difference is the blast transformation. When lymphocytes, they are exposed to certain chemicals like mitogens or so the other bacterial chemicals, their DNA become active and they undergo blast transformation. They become blast cells. For T cells, this happens with mutagens like phytohemagglutinin or concavenaline, con A. B cells, the similar thing happens with bacterial endotoxins like Staphylococcus aureus toxin or Epstein-Barr virus. So this blast transformation happen in T cells and B cells in response to different chemicals. So these were the differences between T cells and B cells. Now coming to another important structure, that is called T cell receptor. B cells on their surface have immunoglobulins by which they encounter the antigen. T cells, they have a structure on their surface that is called T cell receptor. T cell receptor. It is made up of, it is a glycoprotein. It is a glycoprotein and it is present on the surface of T cells. And what it does, through this receptor, the T cells, they recognize an antigen. The antigen is recognized by T cells only through this receptor. Only through this receptor. With the help of some other receptors, CD3 and CD4. They are associated with CD3. They act as an antigen recognition unit and they are analogous to Ig immunoglobulins present on the B cells. So structurally they are quite similar to the immunoglobulins, and they are often referred to as a member of immunoglobulin family because of their similarity in the structure with the B cell receptor. B cell receptor, this is B cell receptor, this is T cell receptor, and they look almost same. They have got one variable region, both of them have got one constant region, and there is a transmembrane region which is embedded into the membrane. And both of them are made up of multiple chains. This is made up of alpha and beta chains, and here also it is made up of light and heavy chains. And what it does, T cell receptor, it recognizes the antigen. And with the T cell, on T cells, other receptors are also present. One is CD4, because this is supposed to be the helper T cells. So this is CD4, or cytotoxic T cells have CD1 on their surface. And this is the antigen presenting cell. It presents antigen to the T cell with the help of MHC molecule. And this way the T cell become active and its cytotoxic activity starts. The type of T cells, we can divide T cells on the basis of their surface markers or on the basis of their functions. Surface markers are receptors and cluster of differentiation. Cluster of differentiation or CD markers, they are surface glycoproteins, surface glycoprotein antigens present on, they are present throughout the body, but they are very important for immune cells because sometimes we have no other way of recognizing them except the cluster of differentiation antigens. For example, CD3 is expressed only on T cells, CD19 is expressed only by B cells, CD64 is expressed by monocytes, CD66 is expressed by granulocytes, CD68 is expressed by macrophages. So with the help of these markers or cluster determinants or cluster of differentiation antigens, we can identify these cells. What type of cell it is? and at what stage of differentiation this cell is. Because these markers, they also change, they not only change with the type of cells, they also change with their development. For example, the blast cells will have some different markers on them. They will not be present on the mature lymphocytes. On the basis of function, the T lymphocytes, they are divided into regulatory T lymphocytes or effector T cells. Effector T cells are the ones which will actually go and kill the infected cells or mount the immune response, like cytotoxic T cells and delayed type of hypersensitivity T cells. Regulatory T cells are those which do not actively take part in the immune response, but they help other cells. They either activate other cells or deactivate other cells. So they are helper or inducer cells, T helper 1, T helper 2, or the suppressor cells. Suppressor cells, they have CD8 markers on the surface, but they suppress the immune response. And there are other T cells which have both CD4 and CD8 markers, and they act as a memory cells. The regulatory T cells, helper cells, there are two types, Th1 and Th2. Th1 secretes interferon gamma and interleukin 2, and this interferon gamma and interleukin 2, it activates macrophages and T cells, promoting cell-mediated immunity. T helper cell 2, it secretes IL4, IL5, IL6, which stimulates B cells to form antibodies, right? So the regulatory T lymphocytes, among them there are helper T cells. Helper T cells are of two types, Th1, Th2. Th1 will promote cell-mediated immunity, and Th2 will promote production of antibodies or humoral antibody by different kind of cytokines. Th1 will release interferon gamma and IL2, and Th2 will release IL4, IL5, IL6, which will stimulate B cells. There are other T cells which are called suppressor T cells, and on the base, effector T cells are delayed type hypersensitivity T cells. They are responsible for delayed type of hypersensitivity. And cytotoxic T cells, they are also called CD8 cells. CD8 because they have CD8 surface marker on their surface. And helper cells have CD4 markers on their surface, right? These helper cells are also called sometimes CD4 cells. CD4 cells because they have CD4 marker on their surface, and these cytotoxic T cells, they are also known as CD8 cells because they have CD8 surface marker. And CD8 marker, the cells with CD8 surface marker, they interact with MHC type 1 molecule, and the cells with CD4 marker, they interact with MHC2 molecule. MHC2 molecules. This we are going to talk about just in a moment. Now, regulation of the immune response, these T helper cells and suppressor cells, if the T helper cells are more active and suppressor cells are decreased, so it will end up into autoimmunity. And if the reverse happens, for example, there are diminished helper cells and increased suppressor cells, it will end up into immunodeficiency. Now, talking something about the B cells and plasma cells. B cells are another lymphocytes, and this B is derived from bursa of fabricus in birds and bone marrow of mammals, because they develop in bone marrow in mammals and in bursa of fabricus in birds. Incidentally, both starts with B. So these are called B cells. They are 5 to 15% of the circulating lymphoid cells, and they have surface immunoglobulins present on their surface. And when they become active, they become plasma cells, and they start secreting antibodies. Otherwise, they do not secrete antibodies. Antibodies are only present on their surface as a receptor. But once they are matured and they become plasma cells, they start secreting the antibodies like IgM, IgA, IgG antibodies. Other receptors which are present on their cells are IgM, IgD, FCR or CR complement receptor or FC receptor. Plasma cells, they do not have these receptors because they do not then get stimulated by the antigen. Their sole function is to secrete antibodies. This is the diagram showing the development of B cells. First, they are pro B cells. They have pro B cell receptors, and then they become pre B cells. Then they become immature B cells. And here, what happens? The clonal selection. The cells which are against the self antigen, they are either killed or their surface antigens are changed, and they come to the peripheral organ, and when they are stimulated by the antigen, they become plasma cell or memory cell, which are the effector B lymphocyte cell, plasma cell and B lymphocyte memory B cells. Plasma cells are the fully differentiated antibody secreting effector cells. They are the final stage of a B cell. They are factories of antibody production, and they are terminally differentiated cells. They die in 1 to 2 weeks after producing a lot of antibodies. They die. And one thing you should always remember about plasma cells is they secrete only one type of antibody. One type of antibody means they will either secrete IgM antibody, IgG antibody, IgD. Not only this, they will secrete antibody only against one antigen, which means all the antibodies secreted from one plasma cell will be exactly same. They will only act against one antigen, not just one antigen. They will act only one epitope against only one epitope of one antigen. What is an epitope? We will talk about in the next lecture. I mean, that is a site on the antigen that will bind to the antibody. So one plasma cell produce antibody that will bind to just one epitope. Other T cells are other cells, I should say, null cells or natural killer cells. Now we don't use the term null cell, we use the term natural killer cells. They lack a phenotypic marker of both B lymphocyte or T lymphocyte. That's why they were called null cells, because they were lacking the phenotypic markers of both the cells. They are of many types. One is natural killer cells and antibody dependent cellular cytotoxic cells. Natural killer cells, because they are not acting against the specific antigen, they just percept whether a cell is normal or abnormal. It can be abnormal because of infection, viral infection or some other infection, or it can be abnormal because of malignancy. So these natural killer cells, they do not act against a particular antigen. They just act against abnormal cells. They do not have that kind of specificity like the T cells have because of T cell receptor, or B cells have because of B cell receptor. They just act against the abnormal cells and kill them. And the function of natural killer cells is immune surveillance and natural defense against virus infected cells and malignant cells. And these cells are also important in killing the tumor cells. Antibody dependent cell mediated cytotoxicity. Some of the T cells, some of the cytotoxic T cells, instead of acting by the T cell receptor, they act by having a antibody receptor on their surface. Normally the T cells have a T cell receptor, and antigen will bind to the T cell receptor with the help of MHC molecule, and they will become active. But some T cells, they have a receptor for FC part of immunoglobulin. So if a cell is bound by antibodies, or even a pathogenic cell is bound by antibodies, these cytotoxic cell will recognize that cell, and with the help of this receptor and IgG receptor, they will recognize it and kill this cell. This type of antibody is called antibody-dependent cell-mediated cytotoxicity. Cell-mediated cytotoxicity. So these were the lymphoid cells. Now coming to the phagocytic cells, there are two types of phagocytic cells: mononuclear macrophages and polymorphonuclear macrophages. Mononuclear macrophages are monocyte and macrophages. Monocytes are present in blood, macrophages are present in tissue. In fact, these are monocytes. When they enter into the tissue, they become macrophages, and both of them are highly phagocytic. They make monocyte macrophage system. The blood macrophages or monocytes, they are the largest of lymphoid cells. In tissue, when they are present in all the tissues. When they are present in lung, they are called alveolar macrophages. When they are present in connective tissue, they are called histiocyte. When they are present in liver, they are called Kupffer cells. These are the modification of these macrophages: Kupffer cells, mesangial cells, microglial cells of the neurons, osteoclast cells in the bones. These are the phagocytic cells, macrophages present in different systems. Okay. The function of the macrophages, it is not only phagocytosis. That is, of course, a very important function of macrophages. Besides that, they also present antigen to the T cells. T cells, they act as an antigen presenting cells, right? And another function is they secrete cytokines like IL-1, IL-6, tumor necrosis factor, and interleukin 12. And these cytokines, they promote innate immune response. And these cytokines result into the stimulation of innate immune response, inflammatory response, including fever. These are the cytokines secreted by macrophages. Macrophages themselves are activated by interferon gamma, which is secreted by the T helper cells. Polymorphonuclear macrophages, they are three types: basophil, eosinophil, neutrophil. Neutrophils are actively phagocytic form. They are present in all acute inflammation. Eosinophils, they are present mainly in tissue, less in blood. They have two types of granules, small and large. And these granules, they contain hydrolytic enzymes, which they can bring out the cell, and they can kill large parasites. So whenever there is a parasitic infection, you will find a lot of eosinophils surrounding that infection, and these cells degranulate their granules on the surface of the parasite and trying to kill it. Another granulocytes are basophils. They are irregular shaped with two lobe nucleus, and one modification, and their granules, they contain heparin, histamine, serotonin, and other hydrolytic enzyme. The mast cells are also a modification of basophils. They are present in tissues, and these mast cells, when they degranulate, they release a lot of histamine and these enzymes, and they can cause anaphylactic reaction. We will learn in details about these things in hypersensitivity. Now, talking something about the major histocompatibility complex, it is also very important. If you remember the T cell receptor, this is the T cell receptor. We have talked about that, and this is present. This is CD4 molecule present on the T cell surface. As I was saying, the T cell receptor is meant to identify the antigen, but the T cell receptor does not identify the antigen alone present when it is present to it. It only identifies the antigen when it is presented by an antigen presenting cell or even a normal body cells with the help of MHC molecule. MHC molecule. Whenever a cell with the help of MHC molecule will present the antigen to the T cell, only then the T cell will react. So then what is this T cell? Oh, sorry, MHC molecule which is present on the surface. There are two types of MHC molecules, class 1 and class 2. In this picture, we are showing you the class 2 molecule. Class 2 molecule is present on immune cells, and class 1 molecules are present in all the nucleated cells. So class 2 molecule is identified by T4 helper T cells, and class 1 molecule is identified by cytotoxic CD8 T cells. So this is a T helper cells or a CD4 cells. It recognizes MHC class 2 molecule by the antigen presenting cells. Similarly, the cytotoxic T cells recognize MHC class 1 molecule. So this is MHC class 1 molecule. This is T cell, CD8 T cell. This is CD8 T, CD4 cells, which is recognizing class 2 molecule. So with the help of MHC molecule, the T cell will recognize the antigen. But it is not that simple, because the T cell receptor recognizes not only the antigen, it also recognizes the MHC molecule, right? So T cell it is not only recognizing this antigen which is presented by the cell, it also recognizes who is presenting this antigen, the MHC molecule. And if this MHC molecule is different, the T cell or the T cell receptor will consider this whole thing as foreign, not just this antigen, but the whole cell, this whole MHC molecule as foreign molecule. So for this MHC molecule become very, very important. Their structure, their genes, they become really important. So if we look at this is class 2 molecule, this is class 1 MHC molecule. Class 1 MHC molecule has one alpha chain. This 3-lobed structure is one alpha chain, and this is beta 2 microglobulin. And class 2 molecule is made up of 2 chains, one alpha and one beta chain. One alpha and one beta chain. This one is made up of one long alpha chain and one beta 2 microglobulin. Beta 2 microglobulin is a different protein. It comes from somewhere else. It is not HLA protein. And there is a portion that is transmembrane segment. And how these proteins, these alpha proteins and beta proteins are synthesized, there are genes for these proteins. They are present on the short arm of chromosome 6. This is called Hla complex or Hla region. In this region, there are 3 genes, A, B, C for class 1 MHC molecule, and there are genes DP, DQ, DR for class 2 molecules. And one thing you should remember, that these A, B, C genes, they are separately can form different alpha chains, alpha or beta chains, and these can form separate alpha and beta chains. So in one individual, we have multiple copy of genes for the production of these alpha and beta chains. Right? On one chromosome, we have 3 for class 1 molecule, and for class 2, we have 3 and for some and 8 for some other molecule. And in one individual, there are 2 chromosomes, one maternal chromosome, one paternal chromosome. So like this, in one human, there are at least 6 classes of 1 MHC, 6 genes for class 1 MHC alleles, and there are more than 8 for class MHC2 alleles. This we are talking about one single person. And when we see in population there are many more alleles present in the population. For example, for class 1 MHC molecule, there are 10,000 alleles present in the population. For class 2, there are 3,000 alleles present in the population. I mean, this is the number that we know or we have yet identified. So in population, these MSC molecules, alpha chain and beta chain, they are present in, I mean, they are highly pleomorphic, highly polymorphic genes. So what is the importance of this? As I said, the T cell receptor does not only recognize the antigen, it also recognizes the MSC molecule. I mean, compare it with, suppose there is a judge who is sitting in a courtroom and a criminal is presented to him with the help, two constables brought a criminal to him, and the judge identifies the criminal and punishes him and whatever he does. So this is this criminal can be a petty criminal, it can be a terrorist, it can be anyone, any type of criminal. So it doesn't matter for the judge. It is his job to see the criminals every day. So suppose one day what happens? This criminal is not brought by the constable. It is brought by two Chinese troops. So what will happen? The judge is going to perceive it as a chaos, a total chaos. Although the antigen is same, the criminal is same, courtroom is same, everything else is same, but this criminal is brought with not with by the constables but two Chinese troops. The judge is going to see it as chaos. He will think that something bad is happening outside. What has happened? As the China has invaded the whole country or like that. So like this, the T cells, when they are presented an antigen and they recognize this MSC molecule, then everything is fine. They will act against the antigen, against the cell. But if the MSC molecule is different, that is present on the antigen presenting cell or the normal cell, it end up into a very, very strong immune response. The T cell mounts a very strong immune response if the MSC is not recognized. So that is the reason why the allogenic MSC molecules, when they are present, the body reacted to as a foreign particle. Even if it is a human cell with different MSC, the immune system is going to react very strongly against it. There are some other non-MSC antigens, minor histocompatibility antigens or normal cellular proteins. So where are they present? Class 1 MHC molecules, they are present virtually on all nucleated cells. I mean the normal body cells, which are non-immune cells, they all have class 1 MHC molecules. So if the class 1 MHC molecule is presenting the normal antigen, normal body antigen, that is good. The T cells won't do anything. But if this class 1 MHC molecule in a normal cell is presenting some other antigen, viral antigen or bacterial antigen, T cell will just kill this cell. Right? These normal body cells, non-immune body cells. The class 2 MSC molecules, they are present only on the cells of the immune system, like macrophages. And these cells are called antigen presenting cells. And when the T cells recognize the antigen, whether it is a foreign antigen or they just get activated, they do not kill the cell because they know these are the immune cells. It is their job to present a abnormal antigen. So these cells are not killed by the T cells. Right? It is like a normal person carrying a gun in his hand, and here it is like a security personnel carrying a gun in his hand as an evidence. Right? So the no action will be taken against these immune cells, and here it will be taken. But here this will activate the immune system. Antigen presentation will activate the immune system to produce cell-mediated immunity. The class 3 molecules, these are the genes also present in the same area on the chromosome 6. They are responsible for the production of complement, some of the complement proteins in the classical pathway like C2, C4, and properdin and factor B in the alternate pathway, heat shock proteins, and the tumor necrosing factor. The role of MHC diversity is important for us, one in the transplantation, This you will study in great deal in when we will talk about the transplantation. Perhaps the pathology of guys will teach you. And for protecting the species from broad number of pathogens. And MSC is also important for some non-immunological phenomena like identification of a person, individual odor, individual susceptibility to different diseases is based on different MSC molecules. So MHC matching is when we try to match the HLA MHC molecules of two person, that is called HLA matching, and we do it when we have to do organ transplant, right, or some for other things. There are many methods of HLA typing: phenotypic method like serology, microtoxicity, or mixed lymphocyte reaction, or there are genotypic methods like PCR for HLA gene sequencing. So by this we can compare the HLA compatibility of two individuals before doing a organ transplant. This can also be used for, apart from tissue transplantation, this can also be used for disputed paternity, anthropological studies, or because some diseases are associated with different variants, like ankylosing spondylitis is associated with HLA-B24, rheumatoid arthritis is associated with HLA-DR4. So knowing about HLA is important not only for transplantation, for other things also. That's it about today's lecture. Uh, if there are questions, one part is still remaining, and that is the cytokines. We will discuss that in some other lecture. So if you have some questions, you can please ask. Yes, so we have another lecture at 2 o'clock, that is antigen and antibody. So we'll meet after 1 hour again. I hope you know that. Okay then.